All right, welcome everybody. So, my name, as is shown on the slides, is Phillip Compeau. I'm a teaching professor at CMU, and I'm an assistant dean working on trying to get SCS, our school, to develop some more initiatives around open online education, which is something that I've worked heavily on outside of my work at CMU. So, I'd be happy to send you some materials there because I have a lot of things that you can learn about for free. But that's not primarily my purpose of chatting to you tonight. I want to take the opportunity to talk to you a little about our undergraduate program. And I put my email, I should say, this direction on the slides. So you can always feel free to reach out to me if you have any questions, and I'm happy to chat to you. So, uh, how I would start today is, well, this is some big news that we had a couple of years ago. We had a couple who donated $25 million to support the future of our department. It's going to support our launch into a brand new academic building. It's like a $400 million academic building that if you come to CMU, our department's going to move there in fall 2027, which is exciting. And I have like three main goals in a talk like this, which is first and foremost to tell you a little bit about what computational biology is. A lot of times I'll talk to people who are interested in hearing more about the field, but very few students will go through high school and have any real sense of what computational biology is, which to me is amazing. And I think I'll explain why that is here shortly. I also talk about how our program works, what makes it unique. And finally, I talk about kind of what is our culture about as a department and how do we try to do our best to support our students and what is it that they do when they leave and these sorts of things. So the first thing I always start with is, what the heck is computational biology? Normally, if I'm in a room, maybe 10% of students who've gone to good high schools will have any real sense of computational biology. And I see a couple of familiar names in the attendees from our pre-college program in computational biology. So that's exciting to see. But still, most people might not have a great sense of what computational biology really is. And so I always try to give a few examples. So the classic problem, I mean, I've talked to students about comp bio because for 15 years, and I've always kind of introduced things with one single problem that's because this figure behind me has been the same figure for like since like the 70s. Methods have changed, but the overarching picture here has been the same for a long time. And this is the problem that really took me from somebody who walked out of a high school biology class saying, I will literally never study that again. I will vow to myself, I have no idea how anyone in their right mind would want to study biology because the material was horrible and the professor was worse. And this problem made me go from someone with that mindset to realize that I was just wrong. And as somebody who loves math and who loves solving problems and computation and things that are cutting edge, I like all those things. And that's what biology is. And here's the picture I would tell you about, which is you probably know what a genome is, the sum total of all your DNA, which is the same basically in all of your cells. You probably have heard of genome sequencing. Maybe if you're watching this, you've actually had your genome sequenced. And the classic problem goes back to the first thing that had its genome sequenced in the 70s. It was a virus that preys on bacteria. Is that you have a lot of copies of your DNA, whatever it is that you want to sequence. In your case, you can sequence your genome by getting a blood sample and isolating the white blood cells, and you have many white blood cells in that sample. They all have basically the same DNA. So you start with multiple identical copies of your genome there at the top. And then there are lab techniques to fragment these this DNA up into smaller pieces. And the reason why is that the laboratory techniques can read DNA, which is an amazing achievement of science because it's submicroscopic. I mean, even an electron microscope, you'd struggle to see the differences between A's, C's, G's, and T's in your DNA. And yet we can do it through as a result of literally billions of dollars of investment into the ability to read DNA. But the weakness, the superpower weakness here is that you can only read shorter fragments of DNA. These are shorter than what real technologies are, but different technologies will give you maybe a few hundred nucleotides, maybe 10,000, 100,000. Depends on the company that you're working with. Regardless, you're not able to read millions of nucleotides at a time. So the best that you can do is read out those fragments. And then you have to put them back together. It's like putting together an enormous puzzle, except it's not really a jigsaw puzzle. You're using overlapping fragments of DNA. Here you'll notice that the orange fragment overlaps with the blue, overlaps with the teal. And at the bottom, you're starting to be able to infer that genome, TGA, GAA, TA, TCA, and so forth. And this is a great problem because, for one, that's a computational question. And there's tons of work that has been done on this for literally 35 years in terms of algorithms, methods for putting these fragments back together. It's still an active area of research. But it also shows the interplay between that critical thing that we would never be able to do this by hand because you're going to have hundreds of millions of these fragments of DNA if you get your genome sequenced. You have to use a computer. You have to use a fast algorithm. You can't just use the first idea that might come to mind. It turns out that that will fail very, very miserably. And you have to use sophisticated approaches to do it computationally, but you also need the data. So there is an interplay here between the fact that the computation can't survive without a laboratory, but the laboratory's purpose is to generate data for us that we can then put together. So you'd say, well, that's good, right? Then we can sequence a genome. Why would we do that? And this is one of the landmark achievements of science in literally human history, is the ability to read one human genome. I think it's as significant or more significant than putting somebody on the moon because, again, of just how complex this is, how tiny that DNA is, how many nucleotides you have to read, and how accurate you have to be. That was a dream of biologists from the time that they started to learn about DNA until it was finally done in around 2001. And that cost $3 billion of federal taxpayer money in the US to actually achieve. And there was a dark secret to it, which is that even though they've spent $3 billion, they only actually had put together like 92% of the human genome. And so they were missing about 8%. And that's because there are repeat regions that are challenging to assemble. The ends of chromosomes are notoriously hard to read, things like this. And so it actually took 21 more years before the first researchers, and one of the leads on this was actually a Carnegie Mellon alum and somebody who's a collaborator of some of our faculty. They actually put together a complete human genome where they were like 100% certain of all of the nucleotides in it, or close to it, 99.999%. And it actually was taken from an individual in Pittsburgh, just like a mile down the street from Carnegie Mellon, which is cool. But you say, why do we sequence genomes? And that's a fair question. So there's two main answers to this. Number one, we want to study life on Earth. And, if you think about the 19th century, biologists did this enormous amount of taxonomic classification and manual labor to try and understand the tree of life. They knew that species had evolved, and they were trying to categorize species based off of properties that they had. And that's a very noble goal. It's this maybe one of like the central goal of all of biology. But when you start sequencing genomes, you get way more information than you are if you're like, well, how many wings does this bug have? How many legs does it have? Right? These types of questions. And those types of questions become ridiculous when you start studying microbes. So when you have to actually look at something under the microscope, a lot of times things will be very, very different, but you can't see that. Right. And so DNA gives you the ability to measure things genetically and have this very rich data set. So you can do things like here on the left. You can, for example, when a pandemic comes along, you can sequence literally millions, tens of millions of viral genomes from patients. And you can use that to understand how is the virus mutating over time, how are new variants arising, and then how do we build that into treatments for those, whether it's vaccines or other types of treatments for patients who have these particular variants. So that was kind of neat. I mean, the pandemic wasn't exactly a fun time, but from the perspective of biology, it was essentially, the first large event, like biological event like this, where something like computational biology was at the forefront of it. And then here on the right, we have probably my favorite figure in biology, which is an evolutionary tree drawn in 2016 by Laura Hug and some colleagues. This is a simplification of the tree because the real tree is very detailed and has more text than really fits on the slide there, or that you could see. And what's interesting about this, and why I think it's such an attention-grabbing, paradigm-shifting image, is that when you look at it, this large purple region that you see up at the top, that's bacteria. And you think, well, where are we? We're in this tiny green sliver called eukaryotes, and somebody's gone in and labeled humans there so that we feel important. Eukaryotes, as you might remember from your probably extremely boring high school biology class, constitute everything that's alive that you have ever seen or ever will see on the planet, pl- animals, plants, fungi, et cetera, et cetera, et cetera. And then a lot of things that you won't see that are actually invisible to you, things like single cell eukaryotes and so forth. Um, so what this is saying is like from the perspective of the actual tree of life formed by, how did they do this? Well, they sequenced the genomes of as many things, they categorized, this is every genome that we can possibly find out there. Let's use an algorithm to build an evolutionary tree. Fortunately, there are textbooks that are that thick on algorithms for building evolutionary trees, and it's been studied for decades. So they just have to take those approaches and then produce this tree. And what this tree tells us is that this is kind of a bacterial planet that you and I just happen to be passing through. From the perspective of genetic diversity, this is the interest of life is not what you probably would think of in terms of like 19th century biology, which is everything that we can see and touch, but it's everything that we can't see and can't touch. And that largely this huge purple part of the tree in the top right is kind of unexplored and misunderstood, right? So we know some things about it, but we're largely just starting to understand what bacteria are all about. And that kind of makes sense. They've been around for a lot longer than we have. They live in places that we cannot live, like in oil wells where it's almost the boiling point, or in deep sea vents or places like this. And they produce compounds like antibiotics that our cells can't make, that we have to borrow. So there's so much to learn. I often say if I had it all to start out over again, there's a really good chance I would just be a microbiologist focusing on computational approaches. We do this for humans though too. And so we sequence a genome for reasons like sometimes it can save a life. And here, this was as of a couple of years ago, this was the world record for the fastest anyone had ever had their life saved because of genome sequencing. So this was in the New England Journal of Medicine. It's the top medical journal in the world. And that's partly because it saved a life and partly because it was the world record for human genome sequencing. There was an infant whose parents took it to the ER, inconsolable crying. And the doctor said, is there any information you can give us? Because it's a baby, right? It's not going to be able to communicate anything. And they said, yeah, well, a few years ago, we had another baby and it had similar types of symptoms, except we took it to the hospital and it got sick and it got sicker. And like a week or two later, it died of a neurodegenerative condition. And so the doctors are well trained. This is a new era. We know that we're in the era of genomics. And the doctors know, well, that means that there's something genetic here, which means it's worth the investment for us to sequence this baby's genome, something which probably cost, I don't know, a few $1,000s. The first one, genome cost $3 billion, and then we've spent a lot of money making that cheaper. And so they say, let's sequence the genome. Now, I always point out to students, you can see down here, it's a little bit small. The blood sample arrives at 5:01 p.m. at the Genome Center, which they were fortunate they were close to. And they start the sample prep, and then they start sequencing. This means fragmenting the DNA and reading it out, those tiny little fragments. That process takes them about 11 hours. So that's just reading all that DNA, and they have fancy machines that can do it very, very quickly and in parallel. So you're reading a ton of fragments simultaneously, but you're reading hundreds of millions of these things. And so then the process starts of the computation, which is how do you put these pieces back together, and how do you compare that against like a healthy human? I shouldn't say normal, let's say healthy human, somebody who doesn't have this condition. This is done in less than an hour. And it's actually, when I teach students at CMU, I teach a first year course introducing students to comp bio that I'll talk about here shortly. We literally studied that algorithm. And that algorithm, when it came out, the algorithm that was used here, it took the amount of time it takes to put the pieces back together from a process of like was taking weeks to something that then could take an hour on a fast computer. And so it shows you the example, like it's a fantastic example of something where the researchers were just trying to get that time down and just trying to devise a beautiful algorithm, but they wind up saving people's lives in the interim, which is that this is somebody who's very sick who needs the diagnosis quickly. And because of a fast algorithm, they get that from weeks down to 50 minutes. And you can learn about that in an undergrad class. Pretty neat. You might say, what was wrong with the baby? Well, the baby had, they found that it was a recessive condition that the parents had, but they each had one copy, and each of these two children had gotten both copies of that recessive allele. And they had a rare disease that wound up giving the baby a mutation in its thymine transporter 2 gene. And you might not know what thymine is. Might sound familiar. Some people might think of it as thymine, which is not quite right, but it's certainly close to thymine, which is one of our building blocks of DNA. But thymine is vitamin B1. So the doctors said, well, what we can do is, if the baby is not great at transporting vitamin B1 in its body, we're just going to give it a superdose of vitamin B1, so it's able to get it around a little bit. It has a defective gene, but it's able to maybe transport it a little bit, and maybe that'll work. So they fill a syringe with vitamin B1, they inject it in the baby, and the baby is 100% fine and is discharged from the hospital the next day. That's an amazing story, right? And it's also a world record, but I should say in the interim, other people have beaten that world record because this is a very active area of research, and universities and genome sequencing centers compete to try and get that time down so they can save lives even faster. I used to talk about CRISPR when I would introduce computational biology to people, and no one had heard of it, first off. And then um, all of a sudden, we started seeing a lot of people had heard of it because for two reasons, really. Number one, we had a Dwayne Johnson movie about it. Now, I've heard this movie is terrible. Don't take my word for it. Feel free to watch it. But my students who have seen it have said it's not really worth watching, but that's good to bring gene editing into the public eye. And also, we had a rogue scientist who created two designer babies without having the parents' permission or informed really consent. So we actually have a couple of humans who are going to be walking around the planet who have had their genomes edited as embryos. Something that's, you can imagine this is a great technology, but do we really have all the ethical guidelines in place for doing something like this? I'm not sure. In the middle of these stories, though, that grab headlines, and CRISPR has won a Nobel Prize, so people have heard of it for this, but in the middle of this stuff that can seem like science fiction, is real science. So people are using gene editing to get crops that are higher yielding to help tackle world hunger, but that are also potentially tastier, um, that are pest resistant, and so forth. At the same time, I also used to tell students, we in the US, we slaughter something like 100 million pigs a year. So every person in the US, on average, eats like a third or a quarter of a pig per year. We also lose 10,000 people a year in the country who are waiting on an organ transplant that they never get. And if you take, for example, if you have somebody who needs a heart transplant, that's a tough transplant to get. If you take a pig heart and put it in somebody who needs a heart, it works perfectly. The problem is that the human immune system goes haywire and you wind up probably dying in a matter of a few hours. So mechanically it works, but then the immune system recognizes it as foreign, no good. It turns out that it's recognizing it as foreign because of the certain genes that are in the pig genome that don't actually affect the pig's life or how it tastes. So you could edit these out, and then when you harvest the pig, instead of that organ getting made into like gelatin or something, it could go and save someone's life. There's a lot to unpack there in terms of ethical, moral considerations that I won't get into, but there have been recent headlines around that. But this is a technology that, number one, has already saved lives, and number two, is going to start saving more in the coming years that we need to start being prepared for. That's been neat because I talked about it as like literally a hypothesized scientific approach a few years ago. And now it's something that's going to be part of modern medicine. Um, we also don't just look at DNA. DNA is the natural thing to start with, but we also look at things like, for example, RNA. RNA allows us to measure the rates at which your genes are getting turned up and down. And we can measure this even in individual cells. That's important because sometimes diseases, for example, constitute not necessarily a mutation in the gene, but the gene doing the wrong thing at the wrong time or something like that. And so these sorts of technologies can produce beautiful plots like this one above me. So this comes from the Chan Zuckerberg Institute. And it's an example of using this technology for a mouse to sample individual cells in the mouse and to measure its rates of at which it's these this soundboard of how the genes are getting moved up and down in terms of how they're expressed, like the rates at which they're getting converted to RNA and protein. Once you do that, you can say, well, if I have a bunch of cells, they have different rates at which these genes are getting expressed up and down. How is it that I could visualize that? And it turns out, well, you just ask people who have been working on this for like 100 years doing statistics, which these days we call machine learning, which these days we call artificial intelligence. But if you take algorithms for that, you can produce beautiful plots like this that are based off of like 19th century mathematics. And here we go. Each colored point is a different type of cell in the mouse, and you can see based that all these sort of differentiate. And that's a beautiful plot on its own. I remember the day I saw it, but it also allows us to do things like, I don't know, why don't we compare a human against a mouse? Why don't we compare diseased mouse against a healthy mouse and start doing this for more and more things? We're moving up the chain in terms of sort of what we can compare from DNA to measuring genes. At CMU, we're proud because we actually have a big chunk of a $20 million NIH grant to build software tools to help biologists who are running experiments be able to make plots like this and do analyses without needing necessarily a degree in computational biology. So here's another way in which computational biology can kind of come to the rescue. In computer science, we have Moore's law, and in drug discovery, we have something called Arum's law. So that's Moore's law backwards. And what it says is, in today's money, we used to spend $1 billion of today's money, and we would get like 100 drugs as a result of that investment. Pretty good. So, I mean, this is 1950. I don't know, a billion dollars of today's money in 1950 was like $50 million, and you'd get 100 drugs. It's pretty neat. These days, the average cost of bringing one drug to market is like $3 billion. So it's gotten way more expensive, even in today's money, to make a medication. And a lot of the reason for this is because kind of the fundamental science hasn't necessarily changed a lot. It's sort of like with drug discovery, people are hunting through the same types of piles for needles in these haystacks. But a lot of these experiments sort of fundamentally depend on understanding what's going on in the cell and how things are interacting. And the central question of biochemistry in terms of what's going on in the cells are like mostly exterior or the outside of your nucleus is mostly proteins. And these proteins, which are doing everything in your cells, are chains of amino acids. Oh, I went forward instead of playing the video. They're chains of amino acids. And if you untangle the protein, it's a chain of amino acids that folds always into the same final three-dimensional shape. Um, as I said, it's like the central question of biochemistry, which is what is the algorithm that nature uses? I call it nature's magic algorithm that always folds this particular protein into the same final shape. This protein is a granulocyte stimulating protein, so it facilitates some of your white blood cells, but its shape is critical to its function. So if it doesn't have that shape, it's not able to bind to the appropriate targets. You think of another molecule coming along and has pockets and things like this that it has to bind to. If it doesn't have that shape, it can't do it. So understanding how it got that shape in the first place, what's this algorithm for taking a string of letters and folding it into that shape, is of fundamental importance. This is why it's a Nobel Prize winning discovery. So this has been an open question since the 60s. And they run contests to see who can come up with the best approximations in terms of algorithms to try and guess what the final shape of a protein is going to be. And a few years ago, AlphaFold, which was a DeepMind Google project, was able to get the best solution that's ever been produced to that. They actually don't have like a perfect solution to it, but they're very, very, very close. It's unlikely anybody will make an improvement as big as what they made. And that was great. It won a Nobel Prize. Also, it just leads to a lot more questions. So what you saw is after they launched AlphaFold, a bunch of companies started up because there's now a bunch more questions. You answer one question and 10 more pop up. That's how science works. And for example, like these proteins are not static things. They have certain parts of them that can be floppier and certain parts that are more static. We need to understand that. How is it that these change over time? Like when they bind to something, sometimes the parts of the protein can move. And if you're just predicting a final shape of the protein, you're not quite getting all that information. So that's then the next question. Then you want to build it into lab research and understand, like, let's build a virtual component that allows all these proteins to communicate with each other. We also look at medical image data, and we're drowning in medical image data. So there's like an unbelievable amount of data worldwide. Like a third of it is healthcare data, and 90% of that is just images of things, CT scans, photos of things like images of this retina over here. And there's only been a small number of cases where people have actually trained an algorithm to beat a doctor's diagnosis. Um, how is it that we can use algorithms, artificial intelligence, and so on to start actually coming up with answers that can lower healthcare costs by producing automated diagnoses and courses of treatment that can free doctors' time and kind of help our system out even further? I mentioned the virtual cell. Probably I should have this slide, one slide beforehand. But if we can do things like understand how proteins fold, we can do things like build a virtual cell in which we understand, like, here are all the proteins. Well, let's allow the simulation to happen, and then we can change the parameters of that simulation, and we can actually have a cell that we study that's not actually a literal living thing. It's just living on a computer. This is a huge area of funding for venture capital money, especially in Silicon Valley. This was all the rage last year of all these different people pitching companies for building an AI virtual cell. It's actually been a problem in like a known problem that we want to solve in biology for a really long time, but we're now starting to get enough computing power in order to address something like that. The other huge direction of computational biology these days is in automating the experiments themselves. So if you've done like a molecular lab experiment in your high school biology class, that's actually really cool because that was definitely not something that we did in my high school biology class. But a lot of times it's based off of a lot of human labor and pipetting and being very, very careful and moving tiny amounts of liquid around from one place to another and following protocols that have to be followed exactly. That's all very important. It's also the type of thing we can just make a robot do. And at CMU, we have actually two automated biological labs that are much faster than a human and much more accurate than a human. So there's no one in this photo because it's like a sterile lockdown environment. And researchers can use all this equipment and just have the robots give them results and often even write code to change the experiments that they're going to run based off those results that they get. So there was always every time I give this talk, this take becomes less controversial. I've shown this slide for many years, since it came out in 2017, where somebody wrote an article saying all of biology is computational. If you are doing biological work, you're almost certainly generating data. The amount of data that we're generating is growing by the minute. And most biological work is generating data that you cannot annotate by hand or look at. It is no longer the 19th century, and it's very much no longer the 20th century. Biology is a computational discipline. That was, it ruffled some feathers in 2017. At this point in time, it's simply a true statement. And that's why I think we're in a great position. Because if you say, what is it that our major really is? I think our major is the life science major of the 21st century. It's an acknowledgement that if you want to study biology, you need to just be doing computational biology. And that's not saying that you shouldn't understand how the experiments work. And that's not saying you shouldn't have to take cell biology and genetics and fundamental courses like this and understand how molecular biology works. All that is absolutely critical. But that this is a field that is driven by data and algorithms in 2025. So how we structured our curriculum was to think about it that way. And I will talk a little bit about administrative details. You can look at individual courses here, and I'll say a little bit about individual courses. But for the most part, we think of what we do as sitting at the center of a lot of different disciplines that we pull from. So our students take biology courses, they take computer science core courses alongside other SCS majors. They take a rigorous collection of math and stats core classes that are the same expectations that other School of Computer Science majors have. And then you did get to take comp bio courses that sort of pull from each of these areas. And I'll talk a little bit about some of those courses. So one course, for example, you can take in your first year at CMU. It's called Great Ideas in Computational Biology. It won the highest teaching award in the college. And I think one of the things that makes CMU unique is if you go somewhere else and you want to take computational biology, they may have really strong graduate programs for this, where people have come in, they already have some training and they've done these sorts of things, or they come in with a lot of computer science background. Um, we are basically trying to do that from year one. And if you see stuff at the undergrad level at other institutions, what you're going to see is you're probably not actually studying computational biology until your junior or senior year. At CMU, we're fortunate to basically have very rigorous math and CS requirements from the first day that you step into the college. And so it means that we can teach real computational biology in your second semester at CMU, which means that you can take 4 years of computational biology coursework. And we have a collection of different courses here. We just added an ethics course. I mentioned ethics. Ethics couldn't be more important to biology in 2025 than it's ever been. We also teach courses like a genomics course that's frankly at a lot of places would be at like a PhD level. But again, one of the big things is you can start taking it in your second semester. The professors here are a little bit kooky, Dr. Van Nostrand, but I promise you he does an excellent job. We also have a lab course that's just available for anybody in the college. It's become popular even among like CS and AI majors because it's a requirement that our CMU students all take a lab class. But uh, our class is really cool because you get to use robots and see lab automation. And it's also all about data. So even though you're learning the lab techniques and seeing how important experimentation is, you're getting a chance to get your hands dirty with it. You're also then at the end of that, you're not just doing the experiments and understanding how they work. You're generating a lot of data and even have a chance to do a project where you write code to analyze the results that you got and get an answer. So pretty cool. So that's become popular just with everybody, not just comp bio majors. A lot of people want to know, can you double major in CB? We're one of 5 majors in the School of Computer Science. Any student can double major at the entire university, not just our school. Any student can double major in any SCS major. So you could be an art major and you could double major in comp bio or CS or human computer interaction. So the answer is always yes, with the disclaimer. I like talking to students early on in their CMU adventure. Not good to know that you want to double major in something in your senior year, but if you talk to me early, the answer is yes. And you can be a double major within SCS too. So we have students who are comp bio majors and then do an additional major in AI, or HCI majors and an additional major in computational biology. You can take any combination of these if you're in our school. So we have this additional major. It's open to everybody at CMU. I think it's a great fit for people like who still want to do a biology major or who are in our science college. I should say there are a few prerequisites, some prerequisites you can see behind my head, and there's a 3.0 QPA, which was what we call GPA at CMU because we have to be different. There's also a chance to take a lighter load here, so you don't have to do all of this coursework, although some students want to. We have a minor and a concentration. The minor is available if you're outside of the college. That's a great fit for, say, bio students who want to take like 5 classes in comp bio. The concentration is available for SCS students. The idea there is we're still going to take 5 classes. One of those classes is going to be a very rigorous machine learning class. And then we're going to give you courses that are a little bit more computationally advanced. So these are for people who are more algorithmically focused and want to study computational biology more from that lens. To join us, I say apply to SCS first choice. So I don't think they typically look at a lot of applicants who have not selected the School of Computer Science first choice. As I said, we have 5 majors in the School of Computer Science, which is cool. We were the first major that was not computer science, that was an option, and now there's 5 total options. You can indicate that interest on your application. You might wonder, what is it that like we're looking for? And that's a great question. As a school, we're looking for, I think, a couple things. Number one, we want a strong quantitative background. So you have to be good at math and you have to show like, that you've been able to kind of max out what your school offers and that you've done everything that you can to take as much math as you can. We also love seeing students who can speak to, here's my background, and then how is it that our program or another major is the right fit for you. Whatever it is that you're interested in studying, how can you speak to that? And then the final thing is, I would say, in our case, what are we looking for? We're looking for all of that, sure, but we're also looking for students who have a passion for biology and in particular a passion for science. I hope it's clear that I have this. And if you're going to study this for 4 years, that's something that I think we really want to see that you can convey. Um, and then the final point of something that I didn't say is computational skills. So those are a plus, right? We still are in an era where um, not ever, we're like minority of universities or high schools in the US offer AP Computer Science A, right? So it's not an expectation that you've done AP Computer Science A, and we understand that we're still kind of, as a country, working out the details on getting computer science education universal at the high school level. Um, you might think, well, I need to code since I was 4 years old in order to be a student at CMU. The professors at CMU, I will tell you, detest that type of thinking. That the only person who can be a computer scientist as an 18-year-old is somebody who has had to be doing this their entire life. And honestly, I would say if we accepted only people like that, it would make for an absolutely miserable environment. Just because it would be too everybody would be too similar. And we're looking for people who have a diversity of backgrounds and interests and things that they want to do. And we have like, it's something like 40% of the class takes an introductory programming class in their first semester because they don't have something to place it out of. And I teach one of those. I teach one that's science oriented, and then there's a class called 15112 that you can take as an alternative as well. So don't feel like you have to have been coding since you were 5. It's not what we're looking for. We're looking for passionate people. And in our case, we're looking for the scientists of the future. So I wanted to have your questions. Thank you so much for taking the time to join me if you're here synchronously. But I do want to say a couple of final points about like jobs and student life and what we offer our students and what we do. I always tell students it's a great meme here. One of the things that's really cool about our program is that it gives you the option to, I already talked about you can use an algorithm to save someone's life, right? And I think that a lot of students, regardless of what you're interested in computing, I think a lot of students really get this that, we've been through a tech era where how beneficial have some of these platforms become to society? And that has become more and more dubious over time. I remember when a lot of these tech platforms were new and they were amazing. And these days, is that true? I'm not sure. Um, Regardless of whether you study comp bio, I think comp bio gives you an avenue for doing things that are like for social good. But we have a lot of different options at CMU of things you can do. And even if you're a computer science major, there's lots of avenues there, especially as computing continues to revolutionize a lot of different aspects of human endeavor. So I think that's kind of what we're looking for too, I would hope, in terms of students, like what they can speak about. We also have fun with our students in all seasons. This is something that I actually launched as a department initiative, not just for our undergrads, but for all of our students. We have a PhD program, we have 2 master's programs, we have like a few hundred students in our department as a whole. But we try to have fun with students. So we have picture days, we have ice cream socials, we go to a local amusement park for Halloween, we go snow tubing in the wintertime, we have a DNA day party where we have a big cookout at the end of the year. Behind me is something called a gobblerito, which is a surprisingly not awful turkey burrito that's offered by a local Tex-Mex restaurant in Pittsburgh that we get catered in and do board games and this sort of thing around Thanksgiving time. Um, One of the other cool things that we do is that we guarantee research to all of our majors. So that's been really neat because all of our students, um, most of our students are interested in research in some way. And we were talking to so many students who were like prospective students who were asked us about research in comp bio that we were just like, yeah, let's just guarantee it to all of our primary majors. So that's been amazing. At the high, like it's been amazing for everybody who's taken part in this. Um, I'll show you 2 really cool stories that are not something that we would expect of everybody who does research for credit, but that have been like our really awesome stories. So Maggie over there on the right won the SCS award for the best honors thesis a couple years ago across all the majors. And she's doing a PhD at Princeton and doing fantastic. And then Daniel here on the left, Daniel did research in the Finning lab and he wound up doing research that was so impactful that they submitted it to Science. And then it was part of a special issue of Science. Essentially, it's being referenced here on the cover of Science magazine, where he was the one of 2 primary authors on the paper. That's impressive because we're pretty sure that had never happened in the history of CMU. Most universities will not have had an undergrad publish a paper in the number one scientific journal in the world. So that's pretty cool. And he's, of course, gone on to fantastic things, doing a PhD at MIT. He's got postgrad fellowships and so forth. We also love connecting students to alums and people doing real work in industry. A lot of times, folks think like, what are the opportunities that are going to be available to me after I graduate? Well, we show you what those are because we have people come to the campus, but we also have started doing like a virtual industry fair in the wintertime. And last year we have like 52 different companies talk to our students, which was really neat because they're all in some aspect of biotech or pharma. Sarah talked to one of those companies called Revilico. I had never heard of the company, but they said, "Hey, can we come to your industry fair?" And I was like, sure. And the next thing she knew is like a few weeks later, she had gotten to another interview. She had a job lined up for them. She has this fantastic job doing like AI for biology, working at this startup that's looking at drug discovery. We also have Megan here. She works at Incitro. That's a company that has multi-billion-dollar contracts from pharmaceutical companies to use like lab automation plus machine learning algorithms in order to find the drugs of the future. Stephen over there on the right is a data scientist at a longevity startup. He's trying to help us live longer and healthier lives, which is something that if you think about making that out as a commercial product, it's probably something pretty valuable if we could all live 10 years longer and stay healthy. Our students also started their own student organization for Comp Bio. They have alum panels, and they're going to have a meeting next week to chat to folks who did internships last summer to find out, all the cool work that people did. They also just get together and hang out and talk about what classes to take next semester. They have research events. They have all kinds of cool stuff that they organize. We sent the first two students to medical school that anyone can remember in like the last 30 years, 30 to 40 years as a school. Um, so this is also another opportunity for, hey, that's my name there behind me. This is another opportunity for people who are like interested in medical careers. A lot of times students will do a biology major and apply to med school, and that's cool. There are a lot of biology students who apply to medical school. And a lot of aspects of medical school is how can you stand out? And I think our major has been an opportunity for students who are interested in medical careers to really stand out and do something different and not just study biology, but understand these algorithms and understand what the cutting edge of the field kind of is computationally, and then take that and apply it to medical work. Here are Zara and Ani. They both recently started medical schools, and we're super proud of them. Um, Finally, yeah, hear from others. Ray and Stephanie Lane were generous enough to give us $25 million. We're moving into a brand new $400 million building. You asked Bill Gates what it was that he would study if he were starting out today. And he said, well, if I could go back and if I were a young person today, I would do AI. That sounds good. I might do energy. Sounds good. And I would also do what he calls biosciences. That's where the frontier of computing can have the biggest impact. Jensen Huang is the NVIDIA CEO. I think we probably heard of NVIDIA. It's the world's largest company. And he says, where's the next revolution coming from? He says he calls it digital biology, but we'll agree that it's the same term. So he thinks computational biology is the next big revolution and the big frontier. How are we going to live longer, healthier lives? How are we going to find the drugs of the future? How are we going to design algorithms for helping us be healthier? And how can we continue to unlock the mysteries of biology that like the biologists of the past would have dreamed about? So that's why we exist as a major. I think we're the best science major in the entire world. If you find any major out there that's better than us, that offers something that we don't, let me know because I want to beat them. I'm happy to take any questions that you have. Thank you so much for joining me. I'm going to stop the recording here and yeah, let's chat.